So I want to start by saying thank you to the organizers. It's an amazing meeting. As an experimental biologist, I feel like I got to peek behind the curtain to see how everything works. So it's amazing. Um, so I'm going to start with an advertisement. I know that you're interested in RNA. So if you're also interested in cancer, do consider joining us in November in Texas. Okay, so the way I like to think about RNA is that most of our coding sequence is pretty conserved, right? And so if you look in monkeys, if you compare that, if you RNA-seq their hearts and ours, or if you compare the brain, the genes that are expressed and also their levels are pretty much similar to ours. And so where does the complexity comes from? And so the complexity comes from the parts that are not expressed, right? And 35% of that are introns. And so splicing is very different. It is something that changed dramatically during evolution. And it's something that's interesting for my lab. And so you've already heard about splicing, but the way it usually works is that some exons are included, others are excluded, and they can give rise to different protein isoforms. And my lab is interested in those isoforms that are sometimes very different. So there's one gene, but it can code to different isoforms that can have sometimes even opposite functions. So one can drive cancer, the other can stop cancer, or can even be resistant to a drug. And so the regulation of splicing here is very interesting and very important for disease. And so many things regulate splicing, but in my lab, we study how chromatin regulates splicing. And so if you're new to thinking about chromatin regulating splicing, then I think it's very intuitive when you look at this scheme that everything is very close together. And we also heard about it, of some of it this morning. And so here is the DNA, and it's wrapped around the nucleosome, and nucleosome tails are modified. And so these modifications are important for polymerase II elongation rate, because nucleosomes are road bumps for polymerase II, and so when chromatin is open, polymerase can elongate faster. And polymerase II is very close to the nascent RNA. And splicing actually takes place, and the spliceosome is depicted here by U1 and U2. So splicing takes place during transcription. And so everything is very close together. Polymerase II is important for splicing. Splicing is important for transcription. And the nucleosome and the tails and their modifications are important for both transcription and directly for splicing. And so there's a very big overlap and a very big interplay between all those things. And so here's my take-home message. And if you remember one thing, and it's going to be this, it's going to be a success for me. And so the way we think about cells reacting to the environment is that we think that chromatin changes. And the way we learned to think about it is that then transcription changes. Now I have more, I have less of a gene, and that's the way the cell reacts to the changing environment. But what I want to tell you that if the chromatin is this radio here, that it doesn't only have one knob that can either take the volume up or down, but it can also change the station. Why just make more or less of something when you can make something different? Now create a different isoform that's going to help us react to this changing environment of the cell. Okay, so the first story I want to tell you about, very briefly, it's about how promoters can be used as regulators of splicing. And so in a high throughput screen that we have done, we have identified P300, and P300 is known to be an acetyltransferase of histones. And so you can imagine it's a co-activator. And so when you want to start expression, P300 comes to the promoter with polymerase 2, opens up chromatin, and then transcription can start. But actually, our work showed that it doesn't only acetylate histones, it can also acetylate splicing factors. And in a way, what we have shown is that promoters are actually a meeting place for mRNA processes, not only transcription, but also splicing. And so the way we think about it is that splicing factors are acetylated and then later on push to a specific splicing of the nascent mRNA. And so this was very interesting for us, thinking about promoters as a meeting place. And essentially, we wanted to ask if promoters are a meeting place, are enhancers meeting place for mRNA processes? And so we started very naively, thinking, so we were working on CD44 gene. And so we thought to ourselves, this is so easy. Let's look at where the enhancer of CD44 is. And so the way we, so we were thinking whether enhancers are a meeting place, and essentially it comes from the fact that enhancers and promoters, right, they're in the same place inside our nucleus in space. And so in today's talk, I'm going to tell you about two stories. One of them will show you how enhancers can regulate elongation rate to regulate splicing, and in the other, how enhancers can actually change chromatin in the exon, in a specific exon, and then change the exon's alternative splicing. And so for the first story, I'm gonna, maybe before starting talking about enhancers, just so we're on the same page, I'll tell you that in order to look for enhancers, you would look for certain characteristics. One of them would be open chromatin, and so places in our genome that are sensitive for DNAs. And so those are going to be similar both in enhancers and in promoters. The other thing that we can look for is monomethyl histone 3K4, which you can see here, which will mark only enhancers and not promoters. And the other is opening of the chromatin by acetylation. And there's a very, very good antibody for histone 3K27. So if you hear a lot about that, it's because we use it a lot to see where chromatin is open. And so those are the three modifications of enhancers, and we're looking for the enhancer of CD44. So essentially, we looked in the genome browser and asked, here is the CD44 gene, where is it? Its enhancer. And so you can use chromHMM that actually takes all the chromatin data and looks for places that are, as I told you, have characteristics of enhancers. And so we looked at some of them, and what we've done to try and ask if this is an enhancer of CD44 is that we tethered a protein, we tethered P300. I actually heard about this system before, but let me just tell you how it works. And so we have a guide RNA. We can have a guide RNA to wherever we want in the genome. And what we bring with it is Cas9, but one that doesn't cut, so it's dead. And it's actually conjugated to whatever enzyme you want. Here it's P300 because we wanted to acetylate histones, open chromatin. And we assume that if we open chromatin in an enhancer, we're going to activate the gene. And so we tethered it to a few enhancers that we thought we identified, but actually we couldn't see any difference. And so we were left with asking, do enhancers regulate splicing? but we couldn't find the enhancer of this gene, and so we had to try a different method. And with enhancers, I think it's easier if you first find an enhancer and then walk to the gene, or at least now it still is. And so we started with an enhancer that was already discovered. And so this is the enhancer of the VGFA, that in a minute I'll tell you what it does. But essentially, VGFA expression goes down during development from lymphoid to B cells. And it was shown that the enhancer is methylated during development. And enhancers are like promoters. When they're methylated, they work less. And so the enhancer is methylated and the expression goes down. And then during cancer, the enhancer is demethylated and there's overexpression of VGFA that is actually happening in most of the cancers, and here it happens in CML, in chronic myeloid leukemia. And so, what is VGFA? VGFA is angiogenic protein. It's secreted from cells, and it's secreted from cells that are in need of oxygen or nutrients, and it's secreted to endothelial cells. It tells them, grow, create a blood vessel that will allow me to get the oxygen and nutrients that I want. And so you can appreciate that in over 90% of solid tumors and also in blood cancers, because the need of vascularization inside the bone marrow, so VGF is overexpressed in all of those. And so I'll tell you very briefly about the splicing of VGFA, and I'm only going to say that there's one isoform that we identified previously, and it's called 189. And it's more potent than the other isoforms of VGF, and this is the one that I'm going to talk about today. Okay, so as I told you, the enhancer of VGF was already identified. So you can see it here in this arrow, and you can see the marks that are already discussed, the monomethyl histone 3K4, the acetylation of K27, and also opening of the chromatin with the DNase and hypersensitive sites. So you can see this enhancer, and to actually put the stamp on it and say that it's enhancer, sometimes you want to know that it's in the same place in space, right, as the promoter, that one can affect the other. And so you can look at this high C, and you can stare at it for a bit, and maybe you can see this dot here that says that the enhancer that is in position plus 157, which means 157 kb away from VGFA's promoter. So the enhancer is actually in the same place in space inside the nucleus as the promoter. What you can also see here in green is that VGF probably has more enhancers than the one that was identified. Enhancers are thought to be working in networks, and changing just one sometimes is not enough, and I think you can see it from our results in a minute. So we started by using cells that the enhancer is deleted using CRISPR. And the first thing we saw, we had two colonies that each of them had a mutation in the enhancer. The first thing we saw is that if you look at the total mRNA of VGF, then it goes down. And so mutations in the enhancer make the expression go down, which is known, and we're not interested in gene expression. We're interested in the splicing. Oops. And so we looked at the splicing, and in both of them, when we had a mutation in the enhancer, we saw less of VGF 189 that I told you about before. Okay, so we want to know whether enhancers regulate splicing. We deleted the enhancer. Now we wanted to activate the enhancer and see whether that will support our observation that enhancers regulate splicing. And so we used, again, the system that I told you about before, when we tether P300 to our the places that interest us, and here we tethered it to both the promoter and the enhancer, and this is the change in gene expression in total mRNA amount, and this is the change in splicing. And so whether we tether VGF to the promoter or to the enhancer, we see a change in splicing, now giving rise to more VGF 189. So mutation less, activation more. So this was interesting. It convinced us that the enhancer had something with the splicing. And we did another experiment that actually helped us trying to understand how the enhancer regulates splicing. And what we've done is essentially we did cheap PCR. So we looked at the chromatin after we tether P300 to the enhancer, and we asked how acetylated is it? And so we tether to the enhancer, so we can see acetylation going up at the enhancer, which is actually a control to see that the system works. But the other thing is that we looked whether tethering to the enhancer also changes chromatin at the promoter region. And it actually does, and it makes sense, right? If they're in the same place in space, then tethering to one place is kind of similar to tethering it to the other place. But another thing that we saw that was very interesting for us is that we saw that chromatin changes inside the VGF gene. And not just inside the VGF gene, but in the same place as splicing changes. And that kind of signaled to us that this change in chromatin inside the gene can be the thing that drives the change in alternative splicing. And so I'm not going to go into that, and we heard a lot about elongation rates. And so we did all these experiments with DRB, trying to understand if indeed the enhancer regulates elongation rate, and whether elongation rate is indeed the thing that regulates VGF, and we found that it was. And then later we also asked what was the thing that is maybe binding the enhancer that is creating this very interesting regulation. And so we used available ChIP-seq data. We're working with K562, which is tier 1 in ENCODE, which makes it super easy. And so we asked from all the genes that were shown to bind, they're binding in K562, which bind both the promoter and the enhancer of VGF. And we could find 3 of those, PML, CCNT2, and MUZ, both binding the enhancer, the promoter, and the enhancer. Here's the VGF, so both the promoter and the enhancer of VGF. And again, I'm skipping a bit, and I'll tell you that we manipulated each of these genes, and we asked, is it really a regulator of VGF-A? And we found that CCNT2 is a regulator of VGF, that it actually changes elongation rate. And so I'll summarize these parts and tell you that we found CCNT2 to bind both the enhancer and the promoter of VGF-A to change elongation rate, and accelerating elongation rate promotes gene expression, but it doesn't only do that, right? Because the radio has 2 knobs. It also changes alternative splicing to then create more of an isoform that is more potent in promoting angiogenesis. Okay, so if I'll also make another summary, then during cancer, the enhancer is demethylated to then create more of VGF-A, and not only more, but more of the isoform that is more potent. Okay, so this was this part, and now to the final part, which is another enhancer, but a different mechanism. And so another story that starts with hematopoiesis, this time differentiating to T cells and talking about TAL1. TAL1 expression goes down during differentiation, and sometimes there are mutations that create an enhancer for TAL1 that, and its overexpression is one of the things that cause T-cell acute lymphatic leukemia. TAL1 is a transcription factor, and its expression is very important during hematopoiesis. So this story is different, and it was actually super interesting for us because the chromatin here is very interesting. So TAL1's enhancer is very close by to the gene. You can see it here. So only 8 kb away from the gene itself. And the gene itself is actually very short. And so the enhancer is also very close to the alternatively spliced exon, which is here, exon 3. And many times when you look at trimethyl histone 3K4, which marks both promoters and enhancers, you will find that it marks the enhancer, and then it will mark the promoter, right? We said they're in the same place in space. But TAL1's enhancer that is created by a mutation is actually thought to be very strong. Some people will actually call it a super enhancer. And super enhancers are so strong that the chromatin goes from the enhancer all the way into the gene and actually marks the entire gene. So it's very broad trimethyl histone 3K4. And it's so broad that it actually opens chromatin so massively that's what makes it a super enhancer. It actually opens all the chromatin and really allows for expression to go up tremendously. And what we initially hypothesized is that this change in chromatin in the exon that is alternatively spliced is actually what's making this enhancer regulate splicing of this gene. And so I'm not going to go into the details, but it is this really broad, very distinct modification that goes all the way from the enhancer into the exon that changes this splicing, and that we've shown it by, you already know all of our tricks. So we deleted the enhancer, we activated the enhancer. We also silenced KMT2B, which makes this enzyme that makes this very broad trimethyl histone 3K4, and we show that indeed this change in chromatin is so massive, all the way from the enhancer into the gene. Actually, the thing that changes the splicing here when we delete the enhancer, we see exon 3 exclusion. When the enhancer is created, is mutated, then we see exon 3 inclusion. And for TAL1, it was known that there are two isoforms, but no one really knew how they differ in function. And so this was very interesting for us. I told you TAL1 is a transcription factor. And when you look at the two isoforms that are created, you can see that both have the domain that makes TAL1 bind DNA. And it actually suggests that maybe each of the proteins can be a transcription factor. And so how are they different as transcription factors? And this is what we asked. And we did two things to address it. One was ChIP-seq, and the other was RNA-seq. And I'll show you very briefly our results. And so we overexpressed each of the isoforms with a tag because we couldn't immunoprecipitate each of them. And we compared it to actually immunoprecipitating both proteins, which was what was known for TAL1 so far. And so you can see in this snapshot this control of binding of TAL1 of both isoforms, and then the short. Here is the long. And you can see that the long actually gives maybe a weaker binding than the short isoforms. And this was very interesting for us. You can also see it here when everything is stacked, that it will center around the binding of both isoforms, and the short is much better in binding in those places than the long isoform is, which was very interesting for us. And this is only the binding, but we actually want to know what are the targets of this transcription factor, not only how strong it actually binds. And so we did RNA-seq for each of the isoforms, and we found that the short is actually has more targets, and it's similar in its impact to both isoforms, and the long actually has less targets. But the most interesting result that we got, and I told you that TAL1 is an oncogene, right? That its overexpression drives cancer. And so we actually saw that for TAL1 long, that it promotes transcription of genes that are important for cell growth. But when we looked at the short, it actually promotes expression of genes that would drive apoptosis, which is the opposite of what is known for TAL1. And so essentially one isoform is a tumor suppressor, the other is an oncogene, which was very interesting for us. We did many more experiments, including in mice, also looking at differentiation during hematopoiesis, and we conclude that the enhancer is when mutated, when there are mutations that create a very strong enhancer, there's a modification that goes from the enhancer all the way to the center of the gene that makes, of course, a very, very strong overexpression of TAL1, but also creates TAL1 long, which is an oncogene. And when you mutate the enhancer, or when you change the chromatin, then you get less of TAL1, but not only less, also more of the short isoform that is actually a tumor suppressor. And so TAL1 used to be thought about as a therapeutic target for T-cell acute lymphatic leukemia, but actually our study shows that you don't need to reduce the amount of TAL1, but rather change the isoforms. So if you make more of the short, then it will actually be a better therapeutic target than just abolishing TAL1 altogether. And so with that, I want to finish with my radio and with planting the idea in your head that now if someone's going to say, oh, when chromatin changes and this gene expression goes down, you should tell them you should also look at other mRNA processes, right? Not only just gene expression. Genes have multiple promoters. They have multiple polyadenylation sites and different isoforms that they can create. So just looking at the up and down doesn't give you the big picture. So keep this radio in mind. And with that, I'm finishing, and I'll tell you that Ahmed Siami, my lab, did the P300 project. The VGFA was done by Sarah Daan, and Aveksha Sharma did the last project that I showed you about TAL1. I want to thank, of course, our funding and you for listening, and I'm happy to take questions.
Thank you for that excellent talk. Any questions, Mary?
Great talk and very convincing experimental data. So I have a question regarding the upregulation of activity of the enhancer using the ECCB. So you use it that has 9 with ECCB attached to it, and you design guide RNAs for particular part of them. Are you doing some kind of screening that identify the best guide for upregulation, or you just randomly choose the guide? Because you need to take into account the nucleosome location, right? All this kind of off-target effects, all the other stuff that should be kind of taken into account. Otherwise, you don't know what if it's come from acetylation or this particular cell, it's come of I don't know, disturbing the CTCF side or somewhere.
It's a very good question. Maybe I can go here and answer. It's a great question about guide RNA off-target effects. And I think that's one of the reasons when you usually do tethering, you don't only use one guide RNA, but rather we use 4. So if I need to tether the promoter, I choose 4 guide RNAs. And then if you think about it and there's an off-target, maybe of one of them, it's only going to be 1/4 of the impact that I'm going to get at the place that I want to tether to. And so we don't try them, we just use 4. It's a different approach. It was actually shown experimentally that it works better to use 4 than just 1.
Yes, thank you. An answer regulated splicing is a very exciting mechanism, and you've shown fantastic examples. I was wondering, because with if you have enough transcriptome and gypsy data on different tissues or cell lines, anyone could investigate and like genome-wide analysis to find more examples. Are there any studies like this?
Definitely. I didn't do a good job giving the background, but there were many genome-wide studies that try to look at exons that are alternatively spliced and ask on the chromatin level how they are different from other exons. And so it's work that's been done. You can look at lists of chromatin modifications that are different. We also know, right, that exons are as long as the piece of DNA that is wrapped around one nucleosome. And so almost all of our exons are marked by nucleosomes. The introns are very different than the exon. So there's a lot of work regarding chromatin and splicing genome-wide. Thank you.
Super cool talk. Thank you so much. So you showed that chromatin dynamics regulate splicing, and from the literature we know that the splicing factors regulate splicing. So have you specifically also studied or looked at the relationship between the chromatin dynamics and the splicing factors?
Thank you. I ignore splicing factors, it's true, because I'm marketing chromatin, but of course splicing factors are expressed differently in different tissues, and they of course are the #1 regulators of splicing. The way sometimes we show that it works is that the chromatin is actually an adapter for a splicing factor. And so you can have really a hand that on one hand is a reader of chromatin that can see a specific modification that will eventually bind to a splicing factor that will in a way translate the way the chromatin signals this axon needs to be spliced as. And so this is the connection, definitely a very strong connection between the chromatin and the splicing. And actually there are splicing factors that can change chromatin. So it goes both ways. Everything is connected.
So I'll sort of continue in that. So this, you showed a beautiful function on the mechanism side. So what you just described is a possible mechanism, but we didn't see data that says this is the mechanism in those cases, right? And another alternative explanation would be dynamics, right? Which people have also shown. So if you're going faster or slower, that's going to basically change the availability of different splice sites that are going to be competed, trying to connect to some of the other talks that we heard earlier. And this is all about competition, right? So this is another way that those marks and the enhancers could be affecting splicing basically indirectly through this. If you look at which one of those, or maybe it's a third one that I don't know about, or
so we definitely, when we study a gene, we check everything. We check whether elongation rate would regulate it. It doesn't always do, by the way. No. So when I showed you here that we found one mechanism, we excluded the other. Well, I didn't show that, but definitely we've done it. And I totally agree. There are different layers of regulation. It's not only chromatin. It's not only splicing factors. Everything works together. That's what makes it beautiful and also complex to study, right? Because it's difficult to tease them apart. Definitely layers. I'm not saying it's only chromatin. I'm not saying it's only elongation rate. Different things.
Back here.
Yes.
Thank you very much. That was very informative, also for someone like me with no biology background. So my question is, for enhancers and super enhancers, since they create such a big change in the chromatin, do you think they could have an effect in genes located far away, or is it really a local effect that we see in specific genes?
Wow, a great question. I think that with the super enhancer that we looked on, with the TAL1 and actually other super enhancers, usually they're close by because they create these broad modifications that go through all the way from the enhancer into the gene. So it's not only to the promoter, but also to the entire gene. And again, imagining that if nucleosomes are road bumps for polymerase II, that they unblock the gene so transcription can go to the end of the gene uninterrupted. And so usually if you look at what we call now super enhancers, they are close to the genes. Their genes are very small, so they're not very big. TAL1 has 4 exons and it's super, and everything is very close together. And so the ones that we know so far, definitely in hematopoiesis, are like that. There are different kinds of enhancer. We don't know all of them now, but maybe some work differently.
Hi, Maayan. I wanted to ask you a question about your very first example of CD44. I think you started by this example and you said that you didn't see any difference in splicing pattern, and then you moved to the GFA. So I was wondering whether you looked at the difference between these two examples and whether, for example, a position of the promoter enhancer, they teach us something on the mechanism why in one case you see difference in alternative splicing, but not in the first example.
Thank you. I think that maybe I made a bit of a salad of the two, and I'm sorry. So for CD44, we did see a change in alternative splicing, and so I didn't go into that. But we wanted to continue thinking about promoters as a meeting place, and we wanted to extend it to find the enhancer of CD44. We weren't able to find the enhancer. Finding enhancers is complicated. People do their life work in finding today, right? Maybe in the future we'll be able to do it faster, but now it's super complicated. And so we couldn't learn about the enhancer of CD44. And so that's why we went into looking first, finding an enhancer, and then looking at splicing. And so that worked better for us.
Great, maybe I'll ask one last question. So kind of going a little bit back to Yossi's question about the mechanism of how some of this works. I think the story you show on elongation rates is really interesting, and I was wondering if you could speculate a little bit on, one, how elongation rates is potentially even changing. So how do you actually get a change in elongation rates, the enhancer difference? And then second, going to how is it actually recognizing different splice sites or actually changing the splicing mechanisms? So I realize that might be speculating a little bit.
So the way we showed it for VEGF2, I didn't get into that, but CCNT2 is actually phosphorylating the tail of polymerase II, and that's how it regulates elongation. So it was known before we started, and so it was only finding the piece of the puzzle of it binding both the enhancer, the promoter, and showing that if you manipulate CCNT2, it will also change splicing. And I think that Alberto Comblit showed that polymerase II slowing can either promote splicing to one side or the other. And the way he shows that is really by looking at splicing factor and thinking about opportunities, about slow polymerase II not giving an opportunity to a weak splice site, and then having a splicing factor that is either an inhibitor of splicing or an activator. And so in a way, I would, maybe if I'll summarize it just to simplify, I would say it's opportunities for splicing factors to bind, right? Because binding is not 0 or 1, it's something in the middle. And so it slowing or making polymerase II faster gives different opportunities for splicing factors to bind, to simplify.
Excellent. Well, thank you so much again.
Thank you.